one aspect of my PhD research, and it's part of a paper that's actually currently under review. So, diversity and biology 101. South Africa is not the rainbow nation for no reason at all. We're well aware of diversity. We've heard a lot of, through the course of the symposium, we've heard a lot of talks on genetic diversity. There's been talks that have focused on species diversity, and thanks to this session, we are now looking at functional diversity. So as part of my presentation, I'm also taking functional diversity and looking at one aspect of it, and that's guild diversity, which is essentially the different feeding guilds that make up a community. And how do we measure diversity? So we can measure it at different scales. So there's alpha diversity, which is essentially diversity measured at the local site or patch level. There's gamma diversity, which is the overall regional pool, and that's based on your kind of sampling size or sampling area. We can also measure changes in diversity, and this is called beta diversity, which is essentially the similarity between alpha and gamma diversity. Beta diversity can be partitioned into two antithetic mechanisms, naming nestedness, which is the change or the loss of diversity resulting in a nested subset of what you started off with. There's also the effect of turnover, which is essentially the replacement of what you started off with originally. We might all be familiar with MacArthur and Wilson's island biogeography theory. This essentially says that large connected islands support higher species diversity compared to small isolated islands. So this is something that you can model. It could potentially be a direct relationship. But what does this mean in terms of functional diversity, or for me, guild diversity? So here we've got our mainland patch, and if I can find the pointer. Mainland patch has got a species diversity of 6. But in terms of guild diversity, it only represents 5 different guilds. Similarly, other different patch sizes have different species diversity, but you'll see that it's not a one-to-one ratio. So yeah, so there's definite differences between species diversity and functional diversity. So we're taking this island biogeography theory and applying it to fragmented habitats. These fragmented forest patches can be seen as islands in a sea of modified landscape. There's particularly the case in southern KZN, where we've got lots of indigenous forest patches, but these patches are surrounded by sugarcane, so lots of agriculture, lots of urbanization. So we kind of developed 4 different hypotheses or predictions. One is kind of purely looking at this island biogeography theory concept, looking at patch size and increasing isolation from mainland habitat patches. So we explored this in terms of our species diversity, overall functional diversity, but also looked at the impacts on individual guilds. We also took this one step further and looked at the configuration of patches. So within a sort of a buffer from a habitat patch, would a sort of clustering of different patches also influence what is within that surveyed patch. We expand further on that by also looking at the land use mosaic, so the specific habitat type, so whether it's, in our case, indigenous forest, thicket dense bush, and then also taking into consideration the land use that forest patch falls within. And then finally looking at change in diversity, so beta diversity, and whether turnover or nestedness is responsible for the patterns of change. So the methods that we used, I used a systematic camera trapping grid. We used that to put together a mammal species list, and this is for each survey patch. In GIS, I quantified patch size, isolation for mainland distances. Within a 1 kilometer buffer of each survey patch, we quantified the number of surrounding satellite patches. We looked at the habitat type, so whether it's lowland coastal forest, scarp forest, or coastal thicket and dense bush, and then also quantified the land use type. So for each of our species, we put together its functional diversity, looking at its activity pattern, whether it's nocturnal, diurnal, cathameral, looked at its size, so this is where its mass in kilograms, looked at its degree of diet specialization, habitat specialization, and also its grouping, so whether it's solitary, lives in pairs, or if it's a group living organism. So over a 2-year period within the southern KZN, so between Umtamvuna Nature Reserve in the south and the Umkomaas River in the north, we covered 157 individual habitat patches, and this covered 96 dense bush and 61 indigenous forest patches. And these patches were located within farms, urban residential areas, but also some of our larger protected areas. So some of the kind of more important information is we had an overall gamma diversity. So this is the total species that we found within our survey area. This is 20 species, and it excludes domestic species. It also excludes extralimital introductions or accidental introductions into the area. And our patches overall were roughly about 27 hectares in size. Each of those had an average of about 5 satellite patches surrounding it. It equated to species diversity of just under 7 and a functional diversity of 4.44. But what did this mean in terms of our predictions for species diversity and functional diversity? So overall, patch size and isolation distance didn't influence either of these measures, but the number of neighboring patches did. And also, interestingly enough, the land use classification influenced. In terms of species diversity, our protected areas had the lowest species diversity. In terms of functionality, our farmland patches had the highest functional richness. For carnivores and insectivores, isolation was an important predictor. So the more isolated a patch was from a mainland site, the lower the carnivore diversity. And then also the clustering, so the number of neighboring patches was important, but this was reliant on an interaction with habitat patch type, so the habitat type. So where there's lots of clusters surrounding a coastal forest patch or lots of clusters surrounding a scarp forest patch, it had a positive influence on carnivores. Herbivores, again, also number of neighboring patches, positive predictor, and then interestingly, land use as well. Our farmland had the highest herbivore diversity, whereas our protected areas had the lowest. And in terms of omnivores and frugivores, patch size, isolation distance, and the clustering of patches had no influence, but it was all down to the specific habitat classification. And scarp forest had the highest number of omnivores and frugivores present within our study site. Species diversity was, or beta diversity for species diversity was described by turnover. So this was the replacement of species, whereas functional diversity was essentially where we found that the overall functionality was as a nested subset of the overall pool that we sampled. So what does this mean? It means that the island biogeography theory, well, one size doesn't fit all. The fragmentation measures such as patch size, isolation distance, and connectivity did not fully explain the diversity measures that we tested. And this is perhaps because our feeding gills varied in their response in terms of fragmentation measures. We can also not exclude the impacts of the surrounding matrix, so the different land use types, and then specifically the habitat type. Although our true forests, as I showed in one slide, are very patchily distributed, and they can be considered these habitat islands in a sea of modified landscapes, the influence of isolation is actually being buffered by thicket and dense bush. So these patches are actually creating corridors and stepping stones between our true forest patches. So we'd like to essentially implore conservation authorities to enact stricter regulations when it comes to clearing these kind of forest-like habitat patches. And this is particularly prominent in southern KZN, where some of our patches where we wanted to go and resurvey, and a lot of the diversity is actually held within our farmland patches, and you get back and that patch has been cleared. And that's because people are increasingly planting macadamia plants. And this is also also means that our environmental impact assessments aren't exactly in place, so we need to kind of think about looking at impact assessments, particularly when it comes to fallow land, land that's been left untouched for longer than 10 years, because it doesn't actually take particularly long for forest patches to regenerate. So that's just kind of the take-home message. And I've got quite a few funders to thank, and we couldn't have done this without private landowners, conservancy, and especially KZN Wildlife that granted us permission to do research in their protected areas. Thank you very much.